Okay, folks, let's get started. Nobody wants to get started. So what's new, right? A couple of announcements. One, I haven't had any takers for the last 2 question and answer sessions on Wednesday, so I decided I'm not going to do them. If you want to come and ask questions, you're always welcome to come. But I just won't plan to be there on Wednesday at 4. So anytime you have questions, please feel free and come by. Second, I've had 2 or 3 people in this class ask me. I teach a class on interview skills in the fall and spring terms. And if you're interested in that, I'll just briefly tell you, it's offered through the University Honors College. It's called BB405H, and it's called Scientists in the Public Eye. You don't have to be a member of the Honors College to take the class. But if you are interested in the class, you need to go to the Honors College to petition to get in. And the Honors College office is Strand 229. What I do in the class is teach students how to do an interview, whether that's medical school, graduate school, dental school, job, whatever. Happy to have you in there. So if you're interested in that, let me know and or let them know, and hopefully you can do that. Okay. Last time I got talking about transcription. I'm going to try to finish it up today. I probably won't make it quite all the way through unless I go twice as fast as I normally do. And I know you guys would like that, right? So, all right. So last time I told you about what I thought was a pretty cool, excuse me, a pretty cool mechanism for terminating transcription. And I unfortunately used the term sequence dependent. And that's really not what it's called. It's called factor independent. And both of them actually mean the same thing. But the name of the termination mechanism is called factor independent. So this mechanism that I described to you on Monday does not require a protein factor for termination to end. In other words, there's no protein that comes along and peels everything apart. The peeling is actually done by this stem loop that I described last time. So it's called factor independent termination of transcription. And I made note of that in the highlights. When I was running through it in my head, I made comment. It is sequence dependent because this sequence has to be there, but it's not typically called sequence dependent. It's called factor independent. Okay. So because of this mechanism, cells have a very efficient and relatively simple way of terminating transcription. And terminating transcription is important because cells don't want to waste energy going too far with making a transcript. Once they finish transcribing the gene, it's really nice to be able to just stop. And so this mechanism allows cells to stop. It turns out that E. coli cells, and this is a mechanism in E. coli, E. coli cells have more than one way of terminating a transcript. So the other way that they use is what's called factor-dependent transcription termination. So it requires a protein factor. And this protein factor that it uses is really cool and interesting. The protein factor that it uses is called Rho, R-H-O. Okay? And Rho actually helps to make everything fall apart. And I'll explain how that works to you in a second. It is a factor that is not absolutely specific. That is, it doesn't cause a transcript to start exactly at the same place. You can see possible places where it stops. And you'll see why that's the case when I describe the mechanism by which it works. Okay? So Rho is a protein that helps prokaryotes to terminate transcription. And it doesn't stop exactly at a specific point. It has what we would describe as preferred places of termination. And like I said, I'll show you why that's the case in a second. Okay. Now, how does Rho work? Well, Rho is a protein that has a very cool property. I always tell this story in this class, and I think it's a very vivid image of climbing a rope in gym class. How many people had to climb a rope to the ceiling in gym class? How many liked doing that? 1 or 2, okay? The first time I did it, I absolutely hated it, and after I got used to doing it, it was kind of fun to do. But I know what the feeling. You just don't want to climb that damn rope one more time, right? Well, Rho is like that, okay? So Rho is a protein that gets on the end of an RNA, once it's come off of the RNA polymerase, and it starts climbing that rope. Now, in contrast to the rope that you climbed, the rope that you climbed in gym class was fixed. It was attached at the top. What if somebody were letting more rope out as you're trying to climb the rope? If you think you didn't like it before, you're going to like it even less now, right? Okay? Well, that's what's happening with this transcript, because this guy is moving along making more RNA as this guy is scurrying up. Okay? So how does transcription termination happen? Transcription termination happens when Rho protein, and notice it's using ATP. It needs ATP for that movement. When Rho protein catches up with the RNA polymerase, guess what it does? It lifts the butt end of it up just like the stem loop did, and everything falls apart. Now, since this is really a race between how fast this guy climbs and how fast this guy moves, it's going to vary then in terms of where it's actually going to stop the transcription. Right? And it turns out, well, when does it catch it? Well, this has to climb faster than this does, than this moves. Okay? It turns out that this guy moves with what we call fits and starts. So RNA polymerization by RNA polymerase does not occur smoothly. It'll go for a ways, it'll sort of slow. It'll go for a ways, it'll sort of slow. It'll go for a ways, it'll sort of slow. And it tends to slow where it hits stretches of GCs. Not surprisingly, those are the ones that have the strongest hydrogen bonds. Okay? So we see places where there are rich GC sequences where this guy is going to take a longer time to get through them, and it's much more likely in those regions that Rho is going to catch up. So we see preferred places where termination occurs, but they're not absolute. In the first case, they were absolute. With the stem loop structure, that is the factor-independent mechanism, once that stem loop formed, it always forms in the same place, everything falls apart. In this case, whenever the Rho protein catches this, everything falls apart. Okay? So it's a pretty cool mechanism. Pretty cool. Make sense? Back in the back.
So how does it ensure that a gene gets transcribed? Like it just seems like
yeah, how does it ensure that the gene gets made all the way through? Well, obviously you want this guy to have a pretty good head start, right? And you don't want to have a lot of slow down sequences in the middle of it. And the answer is that in some cases you may, in fact, make transcripts that aren't full length. Okay? And that's just a trade-off that happens with this. Laurie?
So are the termination zones generally GC-rich?
The termination, the preferred termination sites tend to be relatively GC-rich sites. They do. And no, I don't know how this guy knows to go on to certain transcripts and not others. It's a common question, and it's not completely understood. Rho protein is still being actively investigated for how it actually does what it does, and it doesn't muck everything else up. Back in the back, yes?
when does it start finding the RNA?
Yeah, again, that is under investigation, but it happens once there's enough free space out here for it to do it. It doesn't go on immediately for obvious reasons, because if it did, it would terminate transcription pretty quickly. Yes, back over here. What's that? Okay, so once it catches up, the question was, how does it pull the RNA off? Once it catches up with this thing, it literally does what I said. It lifts the butt end of the polymerase off of the DNA, just like we saw in the stem loop structure, and that destabilizes the whole complex and everything just falls apart. So it doesn't really pull the RNA away. It's just destabilizing the complex is what it's doing. Yes, over here.
What happens to the Rho protein after it's finished?
After it's finished this thing, it just falls apart and goes back and looks for another RNA. Megan?
What was the role of the ATP again?
ATP allows this guy to do the climbing. So when you were climbing that rope, you needed some energy to get up there. It just didn't happen by itself. And so ATP provides the energy for this guy to climb the messenger RNA.
So does it continually use ATP or does it just use one?
So does it continually use ATP? Does it just use one? It's continually using ATP as it's climbing this rope. So what we see from all these things is that, first of all, remember, it took a lot of triphosphates to make the RNA. It's taking some triphosphates to terminate the RNA. And we're going to see when we go to translation, it's going to take a lot of GTP to make the protein. So doing all these functions is very, very energy intensive for the cell, and it's in the cell's interest to maximize its efficiency as much as it can so that it doesn't waste energy. We'll find places where you'll go, wow, why does it do that? Okay? Because it takes a lot of energy to do some of these things, and for reasons that aren't in many cases completely clear, there is a selective advantage for the way the system exists today. E. coli is relatively simple. When we start looking at eukaryotic systems, much more complexity and much more places where you're going to scratch your head and go, wow, why does it waste energy in that way? Okay. So that's termination. So factor dependent and factor independent. This is the factor dependent mechanism. When we talk about inhibition of transcription, there's two different mechanisms I want to describe to you. Inhibition of transcription is useful because if we inhibit transcription, we basically kill a cell. Cell needs RNA to make protein, needs protein to stay alive. So if we stop transcription, we can kill a cell. And that may have very useful purposes for the purpose of, say, killing bacteria. If we can find drugs that stop transcription in bacteria but don't affect our transcription, then we just made an antibiotic that's very, very useful for us. And one of those antibiotics is shown on the screen right here. It's called rifampicin. Rifampicin binds to the alpha subunits of RNA polymerase. Okay? It stops transcription. It does not bind very well to eukaryotic RNA polymerases. And as a consequence, rifampicin, it does give some protection against bacteria. Another transcription stopper is more general, and in fact, it's not specific to bacteria. And that one is known as actinomycin D. Actinomycin D acts by binding to the DNA. So it's actually getting inside the DNA and mucking everything up. And since we have DNA, just like bacteria have DNA, it can cause some issues for us as well as for bacteria. Okay. That's an inhibition of transcription. Now, as I mentioned in class, there are three different main types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. And most descriptions are sort of biased in terms of assuming that you're talking about messenger RNA, but in fact, we need to consider all the different types of RNA. We look at the other RNAs in bacteria. We see, of course, transfer RNA and ribosomal RNA have to be coded for within the genome because, of course, that's where they have to be made from. And interestingly, when we look at the clustering of the genes in bacteria, when we look at the genes for these in bacteria, what we see is that they're clustered. Okay? So the other RNAs, the tRNAs and the ribosomal RNAs, are clustered in one region of the genome, and they're made as one long RNA. So we see the different ribosomal RNAs, and there's their names. We'll say more about those later. We see tRNAs in here. Okay? And this is made as one long complex that is then chopped up into pieces using some enzymes. Okay? Yep. Now, I don't have figures for it, but I will point out to you what we have here. So we saw that tRNA that was in the middle of that, or the tRNAs, there's many tRNAs, that was in the middle of that transcript that I showed you on the last slide. Ribonuclease P, or RNase P as it's called, it cuts and cleaves at the 5' end of transfer RNAs. So we have the 5' end of the transfer RNAs that's there. Ribonuclease III cleaves out 5S, 16S, and 23S ribosomal RNAs. And then after that, those tRNAs have been removed from that transcript. There's some additional modifications that happen to them with some additional enzymes. And one of them is the addition of a sequence to the 3' end of the sequence known as CCA. And I'll show you that in just a minute. Okay? So at the 3' end of all of the transfer RNAs, there's a modification that involves the addition of a CCA at the end. That's the 3' end. So we've got the 5' end fixed. by this ribonuclease P. The 3' end of the transfer RNAs are fixed by several enzymes that are involved in cutting and putting on this CCA onto the end. Okay. Let's see. Actually, I will show you tRNAs when I get later down into eukaryotes, so I'll have a figure for you at that time. But just kind of keep you, I'm sure you know what tRNAs look like. One of the things that happens in RNAs a lot that doesn't happen in DNAs, at least not to any significant extent, is chemical modification. Chemical modification of RNAs, especially in prokaryotes, transfer RNAs, occurs a lot. So what I have on the screen is one modified base inside of transfer RNA. This has been the addition of 2 methyl groups to the amine of adenine. And these changes are happening in prokaryotes. They also happen in eukaryotes. So tRNAs are commonly modified in both prokaryotes and eukaryotes. The nature of the modification, again, is one of those things that's very much under investigation. Why is this modification being done? Why is it necessary? It may, in some cases, be important for flagging that this is, in fact, a tRNA. Okay? If you would make this modification, you could imagine you're going to disrupt some base pairing because that would make a hydrogen bond in base pairing. And so when we look at tRNAs, what we discover is that they have regions that are duplex, but the duplexing is unusual. And so again, these chemical modifications may play a role in altering how the tRNA actually has the shape that it does. Other things that modifications may do may work with what's called the second genetic code. Okay? Now I'm just going to plant that word in your head. I'll talk about the genetic code and translation later. But suffice it to say that there is one mystery about the genetic code, and that mystery may be partly answered by some of these modifications that I'll be talking about. Okay, so modification of bases inside of transfer RNAs, and to a lesser extent, ribosomal RNAs, occurs not uncommonly. Here's a couple more modifications, etc. There's that ribothymidylate. Remember I said that in transfer RNA we can have a ribothymidine? Okay, so that we really should be talking about D-TTP instead of just TTP, because there are places where thymine is attached to a ribose. Okay? All right, that's what I want to say about prokaryotic transcription. As I said, later in the term, probably next week or the week after, I guess next week, I will be talking about gene expression, and we'll come back and revisit how the synthesis of RNA is controlled inside of cells. Okay, well, what I want to say now is something about how eukaryotes do transcription. And there are some significant differences in the process for eukaryotes versus prokaryotes. At a first level, the differences are that eukaryotic transcription is much more orchestrated, much more complicated. Okay? We see many, many proteins involved in the control of the transcription. We'll be talking about those proteins later. But suffice it to say that there's a tremendous orchestration that has to happen. And when I say orchestration, I'm talking about a sequence of events that have to happen in the right order in order for the whole process to occur. Now you might wonder, well, why is eukaryotic transcription so much more complicated than prokaryotic transcription? And part of the answer is the size of the genome. The eukaryotic genome is about 1,000 times longer than the prokaryotic genome on average. So you've got 1,000 times more sequence to find things in. The second reason is that the eukaryotic genome is covered up with proteins. The prokaryotic genome is largely bare DNA in that cell. It's not coated with proteins. So part of what eukaryotic cells have to do is they have to clear out those proteins. And that's a factor for transcription. It's also a factor for DNA replication. Okay. Well, very simple difference, schematic difference between transcription and translation in prokaryotes versus eukaryotes. In prokaryotes, we have no nucleus. So everything happens in the cytoplasm. Transcription happens in the cytoplasm. Translation happens in the cytoplasm. DNA replication happens in the cytoplasm. There's no sequestering of the enzymes for any of these. It's all in the same place. It's all sitting there in what I describe as that cellular soup. Okay? The cellular soup has, which is the cytoplasm, has all those factors already in there for the bacteria. When we look at eukaryotic cells, we have organelles. And the organelles provide some barriers to some of these processes happening. And so when we look in eukaryotic cells, we discover that we have to have some more orchestration just of the processes alone. And one of the things that we discover is that DNA replication and transcription occur in the nucleus. The translation occurs in the cytoplasm. Now, that provides for a decent amount of complexity, partly because we've got to get an RNA out of the nucleus and into the cytoplasm where it can be translated. Now you might wonder, well, why add that complexity? Why not do translation in the nucleus? And the answer to that is quite clear now. The answer is because that the RNAs that are made in the nucleus have to be processed significantly before they're ready to be translated. They have to be processed significantly before they can be translated. If we tried to translate this sequence in the nucleus before it got processed, we would make the wrong protein. Okay? So there's a big difference. The sequestration of the processes. Replication and transcription in the nucleus, translation occurring in the cytoplasm. Okay, so that's a very big difference. Another difference that we see in transcription is that when we look at the bacterial genome, we discover that the genes that are there are very tightly packed in next to each other. Very, very tight. In some cases, a few base pairs separating them. In the case of some bacterial viruses, the genes actually overlap as well. Okay? So we see very tight packing. And when we look at a given messenger RNA that's being made in prokaryotes, it may have multiple genes on one RNA. We don't see that in eukaryotes. In eukaryotes, we see one RNA, one gene. Prokaryotes, one RNA, multiple genes. Okay? The RNA in prokaryotic cells is what we describe as polycistronic. C-I-S-T-R-O-N-I-C. Polycistronic, meaning it has multiple genes on one RNA. Now, when we talk about gene expression, we'll see why that's useful for prokaryotic cells. Another thing we discover in prokaryotic cells is because these processes are occurring in the same place, they can occur at almost the same time. Translation can follow transcription fairly readily. Okay? Can follow transcription fairly readily, so we can actually see both processes occurring as a messenger RNA is being made. In eukaryotes, that's not possible because they're occurring in different places. One in the nucleus, one in the cytoplasm. Another difference that we see in eukaryotes versus prokaryotes is eukaryotes use specialized RNA polymerases to make the different types of RNA. In prokaryotes, we had one RNA polymerase that made all three types of RNA, ribosomal, transfer, and messenger. In eukaryotes, we have an RNA polymerase 1 that works mostly on ribosomal RNAs, an RNA polymerase 2 that works mostly on messenger RNAs, and an RNA polymerase 3 that works mostly on transfer RNAs. So there's specialized RNAs for doing different things in eukaryotic cells. Okay. There they are, 1, 2, and 3. One of the ways in which we can distinguish these different RNA polymerases in eukaryotes from each other is that they have varying degrees of sensitivity to a compound known as alpha-amanitin. Alpha-amanitin is a poison, a big-time poison. It's found in mushrooms. Mushrooms have a name called death cap. And the reason they're known as death cap is if you eat death cap mushrooms that contain this compound, the death cap mushrooms make the alpha amanitin. Okay? If you eat mushrooms that contain this, you are probably dead or you're going to have a liver transplant, if you're lucky. Okay? And there are several incidents per year in the United States and around the world, of course, where people have done exactly that. They've gone out, they've picked some mushrooms, they have cooked them up, they were tasty as all get out, and in the middle of the night, they turn violently ill, and you've got a very short period of time, if that happens, if you're going to survive with a liver transplant. Okay. It happens because RNA polymerase II, which makes the messenger RNA, is exquisitely sensitive to alpha amanitin. It does not take very much alpha amanitin to kill you. Here's the compound. Nasty stuff. Draw that structure for the exam. If you see anything that looks like that, just don't eat it. You know? I know people who are mushroom experts, so I'm no mushroom expert. But in any event, if you don't know what a mushroom is, the safest thing to do is just stay away. Okay. Now, I've alluded to some more complexity in transcription in eukaryotic cells. Yes, sir?
Is that the only source of alpha amanitin?
Is that the only source of alpha amanitin? As far as I know, mushrooms are the only things that make it. And there's only certain mushrooms that make it. Yeah. I don't know of other sources available. Yeah. Bioterrorism toxin there, right? Why do mushrooms make it? They make it as a protective mechanism to keep things from eating them. So as for the same reason that plants make other poisonous substances, it is a protective mechanism. And obviously their own RNA polymerases aren't affected by that compound. Mushrooms are eukaryotic cells. So that is an interesting sidelight to that. Alpha amanitin doesn't have much of an effect on prokaryotic RNA polymerase. So we think about these antibiotics, working on bacteria and not affecting us. Here's one that affects us, doesn't affect bacteria so much. So if bacteria ever become bioterrorists, we're in trouble. They start making this stuff. Yes, ma'am?
Like other toxins, it doesn't matter how much you eat it, little or a lot, it will still affect you the same?
Yeah, is the dose dependent? With anything, of course, dose is dependent, but in the case of alpha amanitin, it doesn't take very much. As I said, it's exquisitely sensitive inhibitor of RNA polymerase II. So it doesn't take much before you're hosed. And there are people who survive it with a liver transplant, but like I said, if you think about it, people on waiting lists for livers or hearts or whatever, and they may wait for months before they get one. And if you have mushrooms tonight, you've got about maybe a 24, 48-hour window, which you might be able to get a liver. So that's one of the reasons it's so deadly and people don't last. Okay. Oh yeah. Here I'm at. All right. So complexity. So we saw in the case of prokaryotes, and I simplified it a bit, but we saw in the case of prokaryotes two primary sequences that were involved in controlling transcription. The -10 sequence, which is the Tata box, and the -35. And to be honest, there are some others. We'll talk about one or two later when we talk about control. But those are the most common ones that we see in prokaryotes, are those two sequences. When we look at eukaryotes, we discover it's a very different world. Okay? First of all, we do see things like Tata boxes. Okay, I'll show you a sequence in a minute that is present. It's not absolutely required. Some genes have Tata boxes. Other genes don't have Tata boxes. The Tata box, instead of being located about 10 nucleotides away from the start, and by the way, that -10 is plus or minus a couple of nucleotides in prokaryotes. Instead of being located 10 nucleotides away, as they are in prokaryotic cells, in eukaryotic cells, the Tata box varies from about 35 to 100 base pairs away. Okay? There's one difference. Second, we see and these are RNA polymerase I, RNA polymerase II, and RNA polymerase III. This Tata box is the only element that they're showing. And by the way, a sequence that's recognized by a transcription factor. A transcription factor is a protein that helps transcription to happen by binding to DNA. A sequence that's recognized by a transcription factor is called an element. A sequence element. So when you hear the term sequence element, they're actually, well, I guess just an element, then what they're talking about is a sequence of DNA that's recognized by a protein that affects transcription in this case. Okay? So an element is recognized by a protein, and that protein has an effect on transcription. Now, there are many different kinds of sequence elements that are located ahead of the transcriptional start site. Okay? Many different elements. I'll show you a couple of these, and I'm going to focus right now on the RNA polymerase II. I won't really say much about RNA polymerase I or RNA polymerase III promoters. They do have some distinct elements of their own as well, including some elements that are inside of the RNA that's being made. Okay? Inside of the RNA that's being made. What I want to say about RNA polymerase II elements is that when we look at them, we see variability in position relative to the start. Variability in position. So one gene may have the Tata box 40 nucleotides away. Another gene may have the Tata box 70 nucleotides away. The other sequence elements may vary anywhere up to 200 or 300 base pairs ahead of the transcriptional start site. So variability. In some cases, we see duplication. We see multiple elements. And then we see something really odd that we don't really see in prokaryotes. Your book alludes to it occurring in prokaryotes, but it really doesn't. The odd thing that we see in eukaryotes that we don't see elsewhere are elements called enhancer elements. Now, enhancer elements, when they were first discovered, were really bizarre. Very, very bizarre. The reason that they were bizarre was it turned out that, first of all, you didn't find them within 100 or 200 base pairs of the transcriptional start. You found them up to several thousand base pairs away. Now, in a prokaryotic system, that wouldn't be practical, because several thousand base pairs away would be in another region for a gene. In eukaryotes, the genes are very widely separated. So there's a lot of space between the genes, and it's entirely possible for an element to go several thousand base pairs and not be in the middle of another gene. They discovered that this sequence can occur multiple times. It can occur ahead of the transcriptional start. It can even occur after the transcriptional start. And the variability of that sequence from one gene to another is enormous, differing by thousands of base pairs. Now, again, we'll talk more about enhancers when we talk about control of transcription. But I will tell you right now that what the enhancers do is they provide what's called tissue specificity. Enhancers are bound by transcription factors that are specific to specific tissues. So you have enhancer elements. They may be active in skin cells, but not in heart cells. So skin cells would have the transcriptional factor that would bind. The heart cells might not have it. That allows for differentiation. It allows for differential expression of proteins. If your skin cells need certain proteins that the heart cells don't need, then they make transcription factors that will make those proteins. Bacteria don't have tissues, of course, so they don't need that complexity that we need of specialization of synthesis of proteins. Clear as mud? Yes? Are these sites not active at all in stem cells? No, you have to keep in mind that any given gene is going to have multiple enhancers, multiple control elements, some of which may be active in certain cells, not in others, some stem cells, not others, et cetera, et cetera. So it's a very, very complicated system. So no, there's no general rule in terms of whether stem cells or differentiated cells or not. So I understand your question, because you're asking about the tissue specificity. And the answer is that even in stem cells, there are some things that stem cells are going to bind and have and others aren't. Good question, though. Yes, Laurie?
So is that maybe why we have a lot of space between genes and you can have a lot of room for these enhancers and various promoters? Yeah, it's a very good question. It's the reason that we have so much space between genes, and it's probably one of the reasons that we do. Yeah, because to make a multicellular organism from a fixed set of genes, we have on the order of about
30,000 genes or so that are there. E. coli has on the order of about 3,000 genes or so, about a tenfold difference. And you look at what happens with that tenfold difference in genes, and you see a tremendous difference in the appearance of a multicellular organism with that of a bacterium. So that's made possible by differential expression, and that differential expression requires some kind of control. So it may very well be that. We'll see that there's other things that allow differentiation of proteins themselves as when we get talking about things in just a little bit. Okay, I promised some we might sing a song today, so I thought we would take a break at this point and sing a song. Would you guys like to do that? I do not have a transcription-specific song. I'm actually working on one. Maybe I'll have one by the end of the term. We shall see. But I thought we'd go back and cover a song that I didn't sing when we did DNA that I think you'll enjoy. If you know the song YMCA, this one I know you know. You have to sing with me. It's a relatively long song, so please sing loud and strong. Phosphates are in nucleotides. I say phosphates. Cover bases inside. I say phosphates. Span the 5 and 3 primes. There's no need to be real mixed up bases. Carry base info you see. I say bases are all complementary. I say bases like A, T, G, and C. They have got to be all paired up. Boom, 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 boom. It's fun to play with some bDNA. It's got a boatload of GCTA. It's got everything a polymerase needs when you melt all the A's and the T's. It's fun to play with some bDNA. It's got a boatload of GCTA. You can make RNAs with a polymerase just by pairing up U's and A's. Proteins full of amino A's, I say proteins come from mRNAs, I say proteins require tRNAs. There is more you need to translate codons. Like our friend UAC, I say codons are all complementary, I say codons have one base wobbly. Now you can go forth and translate. Boom, 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 boom. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. That's enough. Okay. I'm losing my voice. Okay. Let's see. There we go. All right. Hopefully that loosened everybody up a little bit. Now, as I said, eukaryotic transcription requires a lot of orchestration. And so what I'm hoping to show you now is a little bit of that orchestration. With respect to how everything comes together, okay? Well, I'm sorry. Before I do, I want to show you this one thing here. So I said other sequence elements. Here's a couple sequence elements that we see in variable positions ahead of the transcriptional start site. These are not enhancer elements. These are part of conserved sequences around that minus 100 to minus 300 or so region. One's called the cat box. And the cat box got its name by virtue of the fact that, look at there, there's a CAAT that is fairly commonly found. Another head is called a GC box. GC box here, would you expect this would be a place to open up the RNA? No. But it might be very good for recognizing. The protein could recognize and bind there and then open up somewhere else, again, down around the Tata box. The Tata box is useful for peeling apart the strands. Okay. Now for that orchestration that I promised you. So orchestration, there's the Tata box, sorry. You can see it looks very much like the prokaryotic one, TAT-AAAA, okay? And this is the percent of the time that you see each one at that position. So 82%, 97%, 93%, 85%, okay? So it's a fairly well-conserved region for genes that actually have it. Okay, now, the sequence of events that happen in eukaryotic transcription are not something we're going to spend a lot of time on, but I do want to plant the idea in your heads that this order has to be maintained. And Another thing that we see is that transcription in eukaryotes does not simply involve the binding of RNA polymerase to the promoter. Okay? So here's the promoter. This is this region of minus 1 up to, say, minus 300 or so in the eukaryotic cells. The promoter is being bound by proteins that will then help the RNA polymerase to come in. That's significantly different than prokaryotic cells, where the RNA polymerase, with the help of the sigma factor, simply came in and bound to the promoter sequence. Okay? The proteins that allow or facilitate the binding of RNA polymerase have a sort of a standard nomenclature that people use to refer to them. So they have a name that sounds like this. So this guy here is called TF2D. That's a T followed by an F followed by Roman numeral 2, okay, and then a dash D, the letter D. Okay. Now the TF stands for transcription factor. The 2 stands for RNA polymerase 2. So if we were talking about something relative to a transcription factor for RNA polymerase 1, it would be TF1 and then some letter. Okay? TF3, etc. would be for RNA polymerase 3. All right. TF2D refers to this complex, and it's TF2D that's the first transcription factor that binds to start transcription in eukaryotic cells. Okay? So this guy is recognizing some sequences in that minus 1 to minus 300 region of the eukaryotic promoter. Okay? A part of TF2D is a protein. So TF2D is a multiple factors coming together and doing this. Factors being proteins, multi-protein complex. One of the factors is something called the Tata binding protein, or TBP. The Tata binding protein, as its name would imply, recognizes the Tata box and binds to it. So obviously what we're following here are transcription of genes that use Tata boxes. They said not all eukaryotic genes use Tata boxes. In general, the promoters that are more active will have Tata boxes. Those that are less active may not have Tata boxes. Okay, so we see a sequence, and I'm not going to ask you to memorize the sequence. There's just a whole bunch of letters. TF2D binds, TF2A binds, TF2B binds, and then we get a loading on of other factors along with the RNA polymerase. Okay? So there's a sequence that's occurring of the binding of these factors to each other, each of which ultimately is allowing the RNA polymerase to get its start. And then once it's gotten its start, notice that most of the factors disappear and RNA polymerase moves along with the assistance of this protein called T2F. T2F. TF2F, I can't say it. TF2F. All right. So once this guy's going, then we're in the elongation phase, and it's going to go make its RNA. I won't talk here about how termination of transcription happens in eukaryotes. It's not as well known as it's known in prokaryotes. But I will point you to this tail right here that has phosphates on it. This tail with phosphates on it turns out to be an interesting component of the protein. That is, the RNA polymerase protein has a carboxy tail that is full of phosphates. And that tail is actually carrying, I'm doing all this breathing too heavily. That tail is carrying some interesting things with it. We'll see how those things come into play later. Okay? So, we've initiated transcription. We're elongating transcription right now. And termination will eventually happen of transcription. Structural things, there's the Tata binding protein. You can see that one of the things that the Tata binding protein does is it bends DNA. Bending of DNA facilitates the opening of the strands. So the bending of the DNA helps the strands to come apart, and that in turn allows the RNA polymerase to get in and do its thing. There's the Tata binding protein and its interactions with TF2A, for example. Okay. Now, one of the ways in which we see different expression of genes in eukaryotic cells is not unlike some of the things that we saw in prokaryotic cells. Prokaryotic cells were very simple. We saw that one of the ways that prokaryotes differentiated which promoter to recognize depended upon which sigma factor it was making. Okay? And I told you that one of the sigma factors that prokaryotes can make is sensitive to temperature. So we saw a heat shock sigma factor where we didn't see that in normal cells, right? So when we had heat shock, the RNA polymerase in prokaryotic cells was binding to different promoters than it was when there wasn't heat shock. Well, heat shock is something that all cells experience, and eukaryotic cells are not alone in that respect, and they do make other factors that can affect which promoter is being used. So here's a transcription factor in eukaryotes called HSTF, heat shock transcription factor. Like prokaryotic cells, it causes different sequences to be recognized as promoters compared to what prokaryotic cells have. There's a sequence it recognizes if you like sequences. And here is an example gene that has multiple heat shock elements located upstream of it. This is not an uncommon thing in eukaryotic gene organization. We frequently see multiple elements of the same thing, and those multiple elements you might expect would provide more possibilities for the transcription factor to bind and facilitate transcription, and that's exactly why they're there. Remember, we've got a genome that's 1,000 times bigger. We've got a lot more room. We've got a lot of different things. So the more of these elements that we have here, the more likely one of them is going to snag the appropriate transcription factor and facilitate the initiation of transcription. Okay. And let's see, what else do I want to say here? Don't have anything there. In eukaryotic cells, we don't see the organization that we saw in prokaryotic cells. Remember, in prokaryotic cells, we had the ribosomal RNAs and the tRNAs all mixed up on one big transcript. In the case of the eukaryotic cells, we see the ribosomal RNAs on one big transcript, but the regions in between are clipped out. These guys don't become anything. We end up with final ribosomal RNAs, and they have different names because they're different sizes than ribosomal RNAs in prokaryotic cells. Okay? They're made as these guys. And the little red lines that you see on here indicate chemical modification. Remember how I said we had chemical modification that occurred mostly in transfer RNAs, but some in ribosomal RNAs in prokaryotes? We see a decent amount of chemical modification that's occurring in ribosomal RNAs in eukaryotic cells. And that's what those red lines are indicating there, are chemical modifications that are happening. Here's the figure I promised you earlier of the transfer RNA, showing that at the 3' end, there is a processing that happens. So here's the 5' end. At the 3' end, this is an immature transfer RNA. It's immature because it hasn't gained its 3' end yet. There are enzymes that come in and clip off this extension right here, and another enzyme that adds the CCA. And so eukaryotic cells, like prokaryotic cells, have a CCA at the 3' end of their transfer RNAs. This is a mature transfer RNA, and this guy now can get an amino acid attached to it so that it can perform functions in translation. And yes, like we saw in prokaryotic cells, a lot of chemical modification of the nucleotides inside of a transfer RNA. A lot of modification. In fact, you can see evidence of some of the modification. There's a modified base right there, pseudouridine. There's pseudouridine occurring there. There's a T. Okay? Remember, it's a ribonucleotide, so we've got a ribo T in there. And we've got other things, including the Ds that we see out here. So there are some modifications that are occurring to change these bases inside of the transfer RNAs. And let's see. I think that's a good place to stop. I'll talk more about messenger RNA processing next time. That's a fairly involved process.
So heat shock elements, those are they like activated by when heat levels like raise or whatever? Like,
So heat shock elements themselves are just sequences. So if you're talking about the proteins that recognize them, yes, that's true.
okay, so I'll just have a question. One thing I know is that antithrombin deficiency can be caused by severe heat burns.
Uh-huh.
You know? So is that does